Hello, good morning. Myself, I am BSR, botany lecturer. Today, our topic cell division. Quick revision on cell division. Okay, then cell division, different types: amitosis, mitosis, meiosis. First of all, mitosis. Mitosis is also called equational division because there the mother cell can divide it into two daughter cells. I am taking the haplopappus gracilis. In the haplopappus gracilis, in each cell, four chromosomes are present. When mother cell divided into two daughter cells, in that two daughter cells are also same chromosome number is present. So their 2n equal to 4, in the daughter cells are also 2n equal to 4 chromosomes are present. Hence, this division is called equational division. So then, this mitotic division can see in both plant cells and animal cells. But important thing is in the plant, both haploid cells and diploid cells undergo mitosis, but in animal cells, only diploid cells undergo mitosis. Is the important thing. And next, we are discussing about how much time is required for this division. Look at different organisms. You take in human being, 24 hours time is required. In yeast cells, 90 minutes is required. In plants, 16 hours is required. But in that time, 95 percentage of the time undergoing the interface. 95 percentage of the time is required for interface. So the mitotic division can be divided into interphase and mitotic phase. Interphase is the resting period. So interphase is the resting period between two successive cell divisions. So mitotic division, interphase. Again mitotic division, interphase. So first of all, interphase. Interphase may be resting period, but various biochemical changes take place. Based on the biochemical changes, that interface can be divided into G1, S, G2. G1 first. The raw materials for S subphase. What are the raw materials? Proteins, RNA, which are required. So these raw materials are synthesizing in the G1 phase. Those raw materials are required for S subphase. Then when you go for S subphase, this is the important subphase of interface. In this S subphase, DNA content is doubling. There 2C becoming 4C. Only DNA content is doubling because of DNA replication. But chromosome number doesn't change. Chromosome number always same. For example, haplopappus gracilis cell is there. There 2n equal to 4 chromosomes are present. But there the DNA content is becoming from 2C to 4C. Okay. Then G2. In the G2, in the G2, again protein synthesis and RNA synthesis takes place, which is required for mitotic division. So G1, yes, G2. So the main thing S phase where DNA replication takes place. And one more important point, if it is animal cell, centriole duplication also takes place in the S subphase. Okay. Then I'm going to mitotic phase. Mitotic phase can be categorized to karyokinesis and cytokinesis. Karyokinesis is the nuclear division, cytokinesis is cytoplasmic division. The karyokinesis can be categorized into prophase, metaphase, anaphase, telophase. First of all, prophase. Prophase. Generally, in the early prophase, you just observe all the chromosomes are appears in long thread-like. So this long thread-like chromatin material, just initiation starts for condensation. So there is a initiation for condensation. That is the first step. And there the second step, already centriole formation. There the centrioles trying to move towards poles. Centrioles are there, these are moving towards their poles. Not only that one, spindle fibers, initiation of formation of spindle fibers. These spindle fibers are forming by the fusion of microtubules. So initiation of spindle fibers and initiation of centriole movement and initiation of condensation of chromatin material takes place. So that takes place in the early prophase. But slowly at the late prophase, nuclear membrane and nucleolus disappears. Not only nuclear membrane and nucleolus and cell organelles are also disappears during the late prophase. But it is a haplopappus gracillus cell. You observe here 2n equal to 4 chromosomes. 1, 2, 3, 4. But in each chromosome, how many chromatids are there? 1, 2. These two chromatids are formed in the S subphase. You know that one very clearly. These two chromatids are attached to the centromere. On either side of centromere, there a proteinaceous structure is present that is called kinetochore. Very clear. Then after that prophase, metaphase. In the metaphase, mainly two steps. Orientation of chromosomes on equatorial plane and formation of spindle fibers. Orientation of chromosomes on equatorial plane means all the chromosomes are arranged on equatorial plane. Already bipolar spindle apparatus are forming. Look at here. These are the centriole. You know, when the centriole is present, that is the animal cell. Amphiastral mitosis. When the centriole is present, that mitosis is called amphiastral mitosis, that is seen in animals. Whereas when the centrioles are not present, that is called anastral mitosis, that is seen in plant cells. Okay, very important point. Anyway, the spindle fibers are there. These bipolar spindle fibers are attached to the centromere, but to the kinetochores, only to the kinetochores. This is the metaphase. Then when you go to anaphase, in the anaphase, the important thing is these spindle fibers are exhibiting the pressure on centromere. It is exhibiting pressure on centromere. Due to the pressure, the centromere is divided. Centromere is dividing. When the centromere divides, daughter chromosomes are forming. These are the future chromosomes. These are the daughter chromosomes are formed. Later on, these spindle fibers bringing the daughter chromosomes towards their poles. So at one pole, four daughter chromosomes. At another pole, four daughter chromosomes are present. That is anaphase. Then when you go for telophase, telophase is reversible to prophase. It is totally reversible to prophase. It means you just observe their long thread-like chromatin material transformed into rod-shaped structure. Here, rod-shaped chromosomes are transformed into long thread-like chromatin material. Their nuclear membrane and nucleolus disappears. Here, nuclear membrane and nucleolus reappears. All cell organelles are also reappears. That is the reverse to prophase. So at the end of the telophase, in one cell, two nuclei are present. Each nuclei contains 4 and 4 chromosomes. Suppose in plant cell, 2n equal to 14 chromosomes are present. In each cell, 14 chromosomes are present in each nucleus. In that nucleus 14, in this nucleus 14 chromosomes are present. So this is called karyokinesis. Later on, I am going to cytokinesis. The cytokinesis, you just look at here. These are the spindle fibers. These spindle fibers are gathered at the equatorial plane. So all these spindle fibers are gathered at the equatorial plane to form a barrel-shaped structure. This barrel-shaped structure is called phragmoplast. That is called phragmoplast. This is the nucleus. Phragmoplast is formed. Yes, cell organelles reappeared in the telophase. Now the Golgi complex is there. Mainly the vacuoles of the Golgi complex secretes pectin. It secretes pectin. So that is why here a liquid form cell plate is formed. Liquid form cell plate. Now the liquid form cell plate grows centrifugally. Centrifugally means from center to periphery. So which finally moves and finally contacted with the mother wall. Like that a cell plate is formed. Now that cell plate is called middle lamellum. So the first formed wall is called middle lamellum. This is the middle lamellum. Now on either side of the middle lamellum, there is a deposition of cellulose, hemicellulose and pectin. So that is why it is called primary cell wall. It is the primary cell wall. So that side primary cell wall, this side primary cell wall forms. Like that, one mother cell divided into two daughter cells. These two daughter cells are identical with the mother cell. So this is mitotic division. Okay.